Right. I think Dean Riggs gave a very sound introduction of the kind of work that I do. My name is Tawanda. I'm an assistant professor in the Department of Environmental Science and Management, where I teach geospatial courses. So the title of my talk, I think before I get into that, I just want to thank my Department of Environmental Science for the support that they have provided me in terms of acquiring the equipment that I need for my research and teaching. And also would want to thank my family also for supporting me through this journey. So the title of my talk that I want to talk about today is Observing Natural Resources from Space. Yeah. So, a brief introduction. I think most of these things have been mentioned by Dean Riggs, so I don't need to repeat that. I think I'll skip this slide for now. But the other thing that I do is mapping or looking at what is called spatial ecology, where I've been involved in geotagging African elephants in East Africa and mapping their movement patterns, try to understand their habitat utilization, their speed of movements and stuff like that. I think that is one additional thing that I can add to my research profile. So, natural resources is a broad term. So, there are a lot of things. When we talk of natural resources, mainly we talk of soil, water, vegetation, wildlife, and all those kind of things. I just want to talk about one component of my research, which is what is called EBVs, Essential Biodiversity Variables. So these are metrics or measurements that we do as scientists to detect and document the change in biodiversity over space and time. So these are things that include things like that are listed here. We have put things like genetic composition. We also look at species population, species traits, community composition, ecosystem structure, and ecosystem function. So these are some of the things that we can observe from space. So when we're talking of space here, we are talking of anything up from maybe a few meters to about 700 meters up above the Earth's surface. So that is EBVs. So I'm mainly focused on species traits with regard to vegetation, what we often call leaf functional traits. So things like chlorophylls, foliar biochemicals in vegetation, because these ones, they act as a proxy for identifying especially plants that are under stress or that are exposed to any form of disturbance that you can think of. That include fire or any other form of disturbance. So, but however, one challenge with this data is that you need to collect this data on the ground, like what Dean Riggs did explain. So most of this data is to be collected through fieldworks. So you need to do expensive campaigns. Some of these campaigns are very expensive. Some of them, they require a lot of resources that include material, that include human resources, a lot of money also. So remote sensing actually provides us a pathway through which we can complement these field measurements and extrapolate beyond our field measurements. So it provides us an opportunity to look into those areas that we did not sample, because we have got satellites in space. That includes the LEDSAT mission and other satellites that we have in space that can provide us that kind of information. So that is one advantage, or the main advantage of using remotely sensed data. It could be satellites in space, it could be a drone. So now we have drones. With drones, you can mount a sensor. I'll talk about that later on, and then we can map across the entire spatial landscape. So with regard to leaf traits, I just want to give a few of my research that I've done over the few years. So this one is in which I was mapping what is called canopy traits. So basically I was mapping what is called canopy chlorophyll content, and also the amount of nitrogen in plants, leaf nitrogen. So I was using an instrument called DASIS. So DASIS is a hyperspectral instrument which was launched in 2018, and it had for 235 bands. And these bands, they range from the visible spectrum to about 1,000 nanometers, which is our near infrared. So we have about 235 bands, which is a lot of data. So what I did there is that I collected some data in a site in Maine, and then we brought those samples to the lab. We measured the leaf chlorophyll content. We also measured canopy nitrogen. And then we tried to use one of the common, widely used method these days, which is machine learning. We are trying to see if we can use that basis data to map canopy chlorophyll and canopy nitrogen. So that is what is being explained on that part. So the results that we did obtain was with hyperspectral data. Now the challenge is that you've got so many bands, you've got so much data that you need to identify which band combination provides you with the best performance of your model. So basically what we did was, what I did was, I did what is called a band optimization, trying to identify which bands provides me with the best accuracy that I can do. So using that data now, so all these, what we call, these are just coefficient of determinations, which are R squares. So basically what I then observed that with this data set, for canopy nitrogen, I did observe that using a band in the near infrared around 990 nanometers and also at 700 nanometers, I could explain about 76% variation in canopy nitrogen. Yeah? And then for chlorophyll, I did observe that using bands in the red, so the red and also the green spectrum, I could explain about 66% of it. So using those band combinations in a normalized difference vegetation computation, I did manage to map the variation in canopy chlorophyll and canopy nitrogen in this particular landscape. So this is one of the research that I did that I'm still working on. I'm writing this paper and I hope to submit this paper by end of this year or early next year. The other one is, so this is now year numbered. This is data that I did collect with one student in the summer of 2023, and then this data was used by one student by the name of Oren Nadi. Then he used this data to estimate what is called leaf area index. So that leaf area index, that is the amount of foliage that you have in trees. How much leaf material do you have in there? So basically, I did provide this data to this student as a final project that he did for one of my classes that I teach, which is called GSP 330 in mobile mapping. And he used that data and used a multi-spectral instrument called Sentinel-2 to map leaf chlorophyll content. So basically what we did there, we collected data from established permanent plots that have been monitored for over 50 years in the Shorts demonstration farm. And then using an equipment called a plant canopy analyzer, we did manage to measure the leaf area index at those points that are shown on that map. So the good thing also, these results are also, they were also presented at the PCG conference here at Humboldt County last month by this particular student. So what we tried there to do is we're trying several methods. So one of the methods that we tried, we were using spectral bands. Yeah, so probably some of you have heard about what is called normalized difference vegetation index, like NDVI, and other spectral bands that we use. So we're trying bands and indices, and then we're trying to also try to identify a method in which we could minimize that data using a dimensional reduction method to identify which bands are important in estimating leaf area index. So basically what we did, we tried two methods. So we tried what is called partial least squares regression, which is a popular method that has been used since the 1970s. And we also tried random forest, a popular used method. So these are two methods that we tried to use to see which one could give us the best result. So what we did is we ran these models, we did a bootstrapping of these models, ran them several times so that we could compare them. And what you can see here basically is that a method which is called VSEF, which is variable selection for random forest, actually provided us with the best result in estimating leaf area index in the Shorts. And then we use that best performing model to map leaf area index that is shown on that particular map. So this is one method that can be used in mapping EBVs in a forest. And the other project that I've been, so this is a project that I've been doing with my class that I'm teaching now. Is a class which is called GSP 480, which I started teaching this semester. So what we did is that we went to the same short tree farm over a weekend with about 20 students. And then we flew a drone. We actually, my lab, I bought a DJI Mavic 3 multi-spectral drone, which have got about 5 bands. So you've got RGB, near infrared, and red edge. Yeah, and then so we flew that drone and connected it to an MLED unit, and so the drone was receiving what is called real-time kinematic correction, so that you could get very precise location of trees in a forest. Yeah, so that was the correction that we were doing there. Yeah, and then we collected some RGB images, and then we used an open source program called WebODM, which is Web Open Drone Mapping, to generate some orthomosaics and photogrammetry point clouds. And then using that data, we managed to, using the photogrammetry point clouds, we managed to generate what is called the canopy height model, the height of trees in a small area in the short demonstration farm, which is shown in the next picture. Yeah, so this is the height of trees. So what we did observe was that around that, just around the Shorts building there, the maximum height of trees there, or the tallest trees that we could find there was about 42 meters in height. So this is one result that was observed. So I'm planning that to expand on this and try to see if we can write a paper with that student, with this class, to also publish this material and expose students to research and the use of remote sensing. I think because of my time, I've got so many projects that I've been involved in. Yeah. But for now, I think it's important that I end here. All right, thank you so much.